Now we are going to learn organs of the immune system. This is a case about humans, so whatever I'm going to teach you is about your own body. So carefully learn this. So let's start. A number of morphologically and functionally diverse organs and tissues contribute to the development of immune responses. These organs can be distinguished by function as the primary and secondary lymphoid organs. The thymus and bone marrow are the primary or central lymphoid organs where maturation of lymphocytes take place. The secondary or peripheral lymphoid organs include lymph nodes, the spleen, and various mucosa-associated lymphoid tissue or MALT, and such as gut-associated lymphoid tissue and GALT. GALT. These organs provide sites for mature lymphocytes to interact with the antigen. Once mature lymphocytes have been generated in the primary lymphoid organs, they circulate in the blood and lymphatic system. A number of vessels that collect fluid that has escaped into the tissues from capillaries of the circulatory system. Ultimately, this escaped fluid is returned to the blood. So, primary lymphoid organs. Immature lymphocytes generated in hematopoiesis mature and become committed to a particular antigenic specificity within the primary lymphoid organs. Only after a lymphocyte has matured within a primary lymphoid organ is the cell immunocompetent, capable of mounting an immune response. T cells arise in the bone marrow and develop in the thymus. In many mammals, humans, and mice, for example, B cell originate in bone marrow. Thymus. The thymus, site of T cell development and maturation, is a flat, bilobed organ situated above the heart. Each lobe is surrounded by a capsule and divided into lobules, which are separated from each other by a strands of connective tissue called the trabeculae. Each lobule is organized into two compartments. The outer compartment or cortex is densely packed with immature T cells called thymocytes, whereas the inner compartment or medulla is sparsely populated with thymocytes. So immature T cells are called thymocytes. Okay. So both the cortex and the medulla of the thymus are crisscrossed by a three-dimensional stromal cell network composed of epithelial cells, dendritic cells, and macrophages, which make up the framework of the organ and contribute to the growth and maturation of thymocytes. Many of these stromal cells interact physically with the developing thymocyte. The function of the thymus is to generate and select a repertoire of T cells that will protect the body from infection. As thymocytes develop, an enormous diversity of T cell receptors is generated by gene rearrangement, which produce some T cells with receptors capable of recognizing antigen MHC complexes. However, most of the T cell receptors produced by this random process are incapable of recognizing antigen MHC complexes, and a small portion react with combinations of self-antigen MHC complexes using mechanisms that are discussed. The thymus induces the death of those T cells that cannot recognize antigen MHC complexes and those that react with self-antigen MHC strong enough to pose a danger of causing autoimmune disease. More than 95% of all thymocytes die by apoptosis in the thymus without ever reaching maturity. The role of the thymus in immune function can be studied in mice by examining the effects of neonatal thymectomy, a procedure in which the thymus is surgically removed from a newborn mice. Thymectomized mice show a dramatic decrease in circulating lymphocytes of the T cell lineage and an absence of cell-mediated immunity. Other evidence of the importance of the thymus comes from studies of a congenital birth defect in humans, DiGeorge syndrome, and in certain mice, nude mice. In both cases, the thymus fails to develop, and there is an absence of circulating T cells and cell-mediated immunity, and an increase in infectious diseases. Thymic function is known to decline with age. The thymus reaches its maximal size at puberty and then atrophies. This significant decrease in both cortical and medullary cells and an increase in both fat content of the organ. Increase in the total fat content of the organ. Whereas the average weight of the thymus is 30 grams in human infants, its age-dependent involution leaves an organ with an average weight of only 3 grams in the elderly. The age-dependent loss in loss in mass is accompanied by a decline in T cell output. By age 35, the thymic generation of T cells has dropped to 20% of production in newborns, and by age 65, the output has fallen to only 20% of the newborn rate. A number of experiments have been designed to look at the effect of age on the immune function of thymus. In one experiment, thymectomized adult mice were implanted with a thymus from either a 1 day old or 33-month-old mouse. For most laboratory mice, 33 months is very old. So mice receiving the newborn thymus graft showed a significantly greater improvement in immune function than mice receiving the 33-month-old thymus. Next is about bone marrow. Bone marrow is a complex tissue that is the site of hematopoiesis and a fat depot. In fact, the passage of time, fat eventually fills 50% or more of the marrow transplant of bone, marrow compartment of bone. So hematopoietic cells generated in bone marrow move through the walls of blood vessels and enter the bloodstream, which carries them out of the marrow and distributes these various cell types to the rest of the body. In humans and mice, bone marrow is the site of B cell origin and development. Arising from lymphoid progenitors, immature B cells proliferate and differentiate within the bone marrow, and stromal cells within the bone marrow interact directly with the B cells and secrete various cytokines that are required for development. Bone marrow B cells are the source of about 90% of immunoglobulins IgG and IgA in plasma. Like thymic selection during T cell maturation, a selection process within the bone marrow eliminates B cells that possess self-reactive antibody receptors. So despite its key role in humans and mice, bone marrow is not the site of B cell development in all species. In birds, the lymphoid organ associated with the gut called the bursa fabriceus is the primary site of B cell maturation. In mammals such as primates and rodents, there is no bursa to counterpart. This serves as a primary lymphoid organ. In cattle and sheep, the primary lymphoid tissue hosting the maturation, proliferation, and diversification of T cells early in gestation is the fetal spleen. Later in gestation, this function is assumed by a patch of tissue embedded in the wall of the intestine called the ileal Peyer's patch, which contain a large number of B cells as well as T cells. In rabbit too, use gut-associated tissue, especially the appendix, as primary lymphoid tissue for important steps in the proliferation and diversification of B cells. So bursa fabriceus is in birds and Peyer's patch is in cattle, like that. Cattle and sheep. Lymphatic system. As blood circulates under pressure, its fluid component or plasma seeps through the thin walls of capillaries into the surrounding tissue. So in an adult, depending on size and activity, seepage can add up to 2.9 liters or more during a 24-hour period. This fluid, called interstitial fluid, permeates all tissue and bathes all cells. If this fluid was not returned to the circulation, edema, a progressive swelling, would result and eventually become life-threatening. They are not afflicted with such catastrophic edema because much of the fluid is returned to the blood through the walls of venules. The remainder of the interstitial fluid enters a delicate network of thin-walled tubes called primary lymphatic vessels. The walls of the primary vessels consist of a single layer of loosely opposed endothelial cells, even though the capillaries are closed. So we were learning about lymphatic system. The remainder of the interstitial fluid enters a delicate network of thin-walled tubes called the primary lymphatic vessels. The walls of the primary vessels consist of a single layer of loosely opposed endothelial cells, even though the capillaries are closed, blind. The porous architecture of the primary vessel allows fluids and even cells to enter the lymphatic network. Within these vessels, the fluid, now called lymph, follows from the network of tiny tubes into a series of progressively larger collecting vessels called lymphatic vessels. The largest lymphatic vessel, the thoracic duct, empties into the left subclavian vein. It collects lymph from all of the body except the right atrium and right side of the head. Lymph from these areas is collected into the right lymphatic duct, which drains into the right subclavian vein. In this way, the lymphatic system captures fluid lost from the blood and returns it to the blood, ensuring steady state levels of fluid within the circulatory system. In this way, the lymphatic system captures fluid lost from the blood and returns it to the blood, ensuring steady state levels of fluid within the circulatory system. Lymphocytes, dendritic cells, macrophages, and other cells can also gain entry through the thin walls of loosely jointed endothelial cells of the primary lymphatic vessels and join the flow of lymph. The heart does not pump the lymph through the lymphatic system. Instead, the slow, low pressure flow of lymph is achieved as the lymph vessels are squeezed by movements of the body's muscles. A series of one-way valves along the lymphatic vessels ensure the lymph flows in only one direction. When a foreign antigen gains entrance to the tissue, it is picked up by the lymphatic system, which drains all the tissues of the body and is carried to various organized lymphoid tissues such as lymph nodes, which trap the foreign antigen. As lymph passes through the tissues to lymphatic vessels, it becomes progressively enriched in lymphocytes. Thus, the lymphatic system also serves as a means of transporting lymphocytes and antigen from the connective tissues to organized lymphoid tissue, where the lymphocytes can interact with trapped antigen and undergo activation. Then, secondary lymphoid organs. Various types of organized lymphoid tissues are situated along the vessels of lymphatic system. Some lymphoid tissue in the lung and lamina propria of the intestinal wall consist of diffuse collections of lymphocytes and macrophages. Other lymphoid tissues are organized into structures called lymphoid follicles, which consist of aggregates of lymphoid and non-lymphoid cells surrounded by a network of draining lymphatic capillaries. Until it is activated by antigen, a lymphoid follicle called a primary follicle comprises a network of follicular dendritic cells and small nesting B cells. After an antigenic challenge, a primary follicle becomes a larger secondary follicle, a ring of concentrically packed B lymphocytes surrounding a center, the germinal center, in which one finds a focus of proliferating B lymphocytes and an area that contains non-dividing B cells and some helper T cells, interspersed with macrophages and follicular dendritic cells. Lymph nodes and the spleen are the most highly organized of the secondary lymphoid organs. In addition to lymphoid follicles, they have additional distinct regions of T cell and B cell activity, and they are surrounded by a fibrous capsule. Less organized lymphoid tissue, collectively called mucosa-associated lymphoid tissue, are found in various body sites. So, MALT includes pairs, patches in the small intestine and tonsils and appendix, as well as numerous lymphoid follicles within the lamina propria of intestines and the mucosa membranes lining the upper airways, bronchi, and genitourinary tract. Then, lymph nodes. Lymph nodes are the sites where immune responses are mounted to antigens in lymph. They are encapsulated bean-shaped structures containing a reticular network packed with lymphocytes, macrophages, and dendritic cells. Clustered at junctions of the lymphatic vessels, lymph nodes are the first organized lymphoid structures to encounter antigens that enter the tissue spaces. As lymph percolates through a node, any particulate antigen that is brought in with the lymph will be trapped by the cellular network of phagocytic cells and dendritic cells. The overall architecture of a lymph node supports an ideal microenvironment for lymphocytes to effectively encounter and respond to trapped antigens. Morphologically, a lymph node can be divided into three roughly concentric regions: the cortex, the paracortex, and the medulla, each of which supports a distinct microenvironment. The outermost layer, the cortex, contains lymphocytes, mostly B cells, macrophages, and follicular dendritic cells arranged in primary follicles. After antigenic challenge, the primary follicle enlarged into secondary follicles, each containing a germinal center. In children with B cell deficiencies, the cortex lacks primary follicle and germinal centers. Beneath the cortex is the paracortex, which is populated largely by T lymphocytes and also contains dendritic cells that migrated from tissues to the node. These dendritic cells express high levels of class II MHC molecule, which are necessary for presenting antigen to Th cells. The medulla is more sparsely populated with lymphoid lineage cells, and of those present, many are plasma cells actively secreting antibody molecules. When as antigen is carried into a regional node by the lymph, it is trapped, processed, and presented together with class II MHC molecules by dendritic cells in the paracortex, resulting in the activation of Th cells. So the initial activation of B cells take place within the T cell rich paracortex. Once activated, Th and B cells form small foci consisting largely of proliferating B cells at the edge of the paracortex. Some B cells within the foci differentiate into plasma cells secreting antibody. These foci reach maximum size within 4 to 5 days and antigen challenge. Within 4 to 5 days of antigen challenge, a few B cells and Th cells migrate to the primary follicles of the cortex, where cellular interactions between follicular dendritic cells, B cells, and Th cells take place, leading to development of a secondary follicle with a central germinal center. Some of the plasma cells generated in the germinal center move to the medullary areas of the lymph node and many migrate to bone marrow. So, afferent, that means incoming lymphatic vessels pierce the capsule of lymph node at numerous sites and empty lymph into the subcapsular sinus. Lymph coming from the tissue percolates slowly inward through the cortex, paracortex, and medulla, allowing phagocytic cells and dendritic cells to trap any bacteria or particulate material carried by the lymph. After infection or the introduction of other antigens into the body, the lymph, leaving a node through its single efferent outgoing lymphatic vessel, is enriched with antibodies newly secreted by medullary plasma cells and also has an approximately 50-fold higher concentration of lymphocytes than the efferent lymph. Some of the increase in the concentration of lymphocytes in the lymph leaving a node is due to lymphocyte proliferation within the node in response to antigen. Most of the increase, however, represents blood-borne lymphocytes that migrate into node through the walls of postcapillary venules. These venules are lined with usually plump endothelial cells that give them a thickened appearance, and they are called high endothelial venules. The high endothelial venules are important because most of the lymphocytes that enter the node arise by passing through the specialized endothelial cells of the HEV by extravasation mechanism discussed. So, a proportion of the lymphocytes leaving a lymph node have migrated across this endothelial layer and entered the node from the blood. Because antigenic stimulation within a node can increase its migration tenfold, the number of lymphocytes in a node that is actively responding can increase greatly and the node swells visibly. Factors released in lymph nodes during antigen stimulation can thought to facilitate this increased migration.